The process of making you who you are is all about how to make proteins, because when you look at everything that you are and everything that you do, it's all based on proteins. The thing is, though, those proteins are built based on the genetic code that's inside the nucleus of every single one of your cells. So your phenotype comes from your genotype. But how does one thing become the other? That's the process of gene expression that we have to talk about. And it happens in two main stages. The first one is transcription. And it's called transcription because the message that's stored in DNA must be transcribed into RNA so that it can go to the cytoplasm where the protein synthesis actually takes place. You're not going to move the DNA to the cytoplasm. You move a piece of the DNA or a copy of it into the cytoplasm in order for it to be translated. Translation, because you're going to change the message from genetic message, both DNA and RNA are genetic messengers, into a different kind of macromolecule called proteins. How does this process actually happen? Transcription will happen in the nucleus with the help of a lot of enzymes, which will make the RNA. In eukaryotes, the RNA must be processed in what's called Post-Transcriptional Processing. The mature messenger RNA will exit the nucleus through the nuclear pores and go to the cytoplasm, where a ribosome, which is made of ribosomal RNA and proteins, will work with the help of a lot of other proteins and enzymes to translate that message. And the transfer RNA, yet another kind of RNA, is going to be picking up the amino acids and putting them in the correct sequence inside the ribosomes. That process is then followed by post-translation modification of proteins, especially in eukaryotes. Let's talk about how each one of these steps takes place. It all starts with Rna Polymerase, which reads the DNA in a 3 to 5 direction, building the RNA molecule in a 5 to 3 direction. It does the job that helicase, topoisomerase, and DNA polymerase does doing DNA synthesis. It uncoils, unzips, and copies the DNA according to base pairing rules to actually create the RNA-DNA hybrid. Eventually that disconnects, and only the RNA will exit the RNA polymerase. There are several kinds of RNA polymerase because there are different kinds of RNA. The one that actually makes messenger RNA, which contains the message to make proteins, it's called RNA polymerase type 2. Other RNA polymerases make other kinds of RNA, which we'll talk about at the end of the video. The RNA polymerase will then create this transcription bubble, and only one strand of DNA would actually be inside the transcription bubble. We call that the template strand. And the template strand is used to make the RNA message in accordance to the base pairing rules. So, for example, if you have TAG, GTT, in the template strand, you're going to make the exact opposite, which is AUC, CAA in the messenger RNA. Remembering that you substitute T's when you look at the RNA because they're more stable if they're going to be unpaired, as they are in the messenger RNA molecule. But notice something interesting, that the strand that's actually used for transcription has the anti-sense, or the opposite sense of what you actually want. If you compare the non-template strand, which is the one that's not read, with the actual message that's in the RNA, you will see that it's very similar. ATC, AUC. Kind of like the same thing where you would substitute it for T. And that's an interesting thing about the DNA code. It is the template strand that's actually transcribed, so you can go back to get the actual code that's in the non-template strand. What does that mean for the DNA code? Only half of the hubble helix actually has coding material. The other half is only there to be the antisense, so that when doing transcription, it can serve as a template to build the molecule that you want backwards. It's all because the RNA polymerase, like DNA polymerase, finds it easier to copy by simply using the base pairing rules and seeing what needs to pair with this specific nucleotide we're talking about here. This means that if the non-template strand is used, you're going to get the entire wrong code instead of getting the code that you actually want. While we're on that page, since the code depends on the triplet code that we talked about in the other review video, it's very important to start copying from the right place. If you start the actual transcription in the wrong place, you get a completely different message and a completely different set of amino acids set up from the actual code, which means the RNA polymerase needs to know where the beginning of the RNA transcript actually is. And the ribosomes, which are actually translating it, also need to know exactly where to start from. This is made possible because every gene in the body will have regulator sequences upstream from the actual part of the DNA that needs to be transcribed, which is called the transcription unit. These promoter sequences will actually mark the starting spot for the transcription process and regulate whether or not the transcription is actually happening. Because you don't want to be transcribing all the time. You only want to transcribe the protein when it's needed. So the cell will only do that in response to chemical messages from the cytoplasm that basically say, hey, we need that protein. When that message is active, you're going to do the actual transcription process, which includes initiation, elongation, and termination. During initiation, a set of transcription factors, which are basically those chemical messengers that come from the cytoplasm, will attach themselves to the promoter sequence of the gene, which is not actually translated, but actually allows the gene to be translated. Once those things are connected to the actual promoter box, which in humans has a lot of adenines and thymines repeated over and over again, over 25 to 35 base pairs long sequence, which we'll call the theta box. When those transcription factors connect there, then the RNA polymerase can finally connect to the DNA and start the process of transcription. And it will work downstream from that point until it reaches the transcription termination site, when it will actually release from the DNA and know that it's completed the process of transcription. That means that without the transcription factors and without the promoter sequence being available and actually activated, no protein synthesis would actually take place, because RNA polymerase will not be able to transcribe the message in the first place. The message that's actually transcribed is called a transcription unit. But if you actually look at the structure of the transcription unit, you will see that it has a lot of information inside. It includes what we call the cap, an upstream untranslated region, the coding sequence, the downstream untranslated region, and a poly-A tail signal. All these things will be important in the preservation and functioning of the messenger RNA molecule, as it does its job of sending the message to the cytoplasm to actually make the protein. And then, if you actually get the coding sequence, which is what actually matters, what actually includes the information to make the protein, you will see that it actually has a bunch of introns and exons. Introns are pieces of the message that do not actually code for the protein, and they must be removed before the protein actually can be made from the messenger RNA transcript. This leads us to the idea of RNA processing, which in eukaryotes, because the DNA looks like this, all RNA must be processed. In prokaryotes, you don't really have all of these introns that need to be removed, so they skip this step. But in prokaryotes, special molecules called SNRPs, which are made of a combination of proteins and yet another different kind of RNA called small nuclear RNA, get together to form molecules called spliceosomes. And the spliceosomes will recognize intron boundary sequences at the edges of the introns and use that to remove the introns from the actual code, leaving only a mature RNA that has the exons only that need to be translated into the protein. But since the spliceosomes can be made of different pieces or different SNRPs, different spliceosomes will recognize different intron boundary sequences and remove different introns, which means it's possible to make many proteins from the same gene, depending on which introns you leave behind. This helps suggest why we have introns in the first place. One of the reasons is that we can make many proteins from the same gene depending on which ingredients we leave or take out. Also, because some proteins can't be made until the introns are taken out, this serves as a method to actually control the gene expression. Also, because you are adding distance between the actual pieces that matter, you're actually going to make this gene more resilient to crossing over events, where the bigger the distance between the genes pieces, the higher the likelihood that the pieces will cross over together. And finally, because the introns are not actually coding sequences, they will increase the chances of you not having a mutation affect the actual protein. Since if the mutation happens in an intron, they're going to be deleted anyways, so it won't matter. Spliceosomes will remove the introns, and then an enzyme called ligase will stitch up the pieces of the messenger RNA together to make the final mature messenger RNA that's ready to be sent out of the cell. But yet another step needs to be made to make sure this RNA is ready. You must add the 5' G cap, which is basically a molecule of GTP, a guanine triphosphate or nucleoside triphosphate that's added to the beginning of the messenger RNA and is going to have a role to facilitate the initiation process of translation later in the cytoplasm. You're also going to add a poly-A tail to the end of the message to actually protect the message from degradation. When the messenger RNA actually gets to the cytoplasm, enzymes are going to be trying to look for messenger RNAs to destroy them. Otherwise, the messenger RNA will always be around and the protein will be made forever. In order to control that, the enzymes destroy messenger RNAs so that only the proteins which are constantly needed can actually be produced. The poly-A tail, as well as the upstream and downstream untranslated regions, serve the purpose to help protect the actual coding sequence from degradation so that this messenger RNA can last longer. The downstream untranslated region also includes the poly-A adenation signal that tells the cell where to add the poly tail so it's not accidentally added to the wrong end. It needs to be added to the 3 end of the molecule. Split up sequence also helps the ribosome attach to the actual messenger RNA. So that's how the final mature RNA looks like. It will have a guanine triphosphate cap, followed by the upstream region, the actual codons which are left, and the downstream region, and the poly A tail. Notice also that every single coding sequence will start with the start codon, or the codon that means this is the first amino acid of the sequence. For every single protein in eukaryotes, that is AUG, which stands for methionine. So in addition to the things we talked about in the previous video about what all proteins have in common, all eukaryotic proteins they will start with the same amino acid. And then they will also end with the stop codons. And there's only 3 different kinds of stop codons that can be used. All of them mean the same thing. This protein is done. UAA, UGA, and UAG will all stand for that same thing. By the way, if a mutation happens on either the start or stop codon, that will really deeply affect the protein synthesis mechanism, because the protein might not even start, or the ribosome might not know when to actually finish the protein making process. And that will definitely affect whether the protein is made successively or not. Other mutations might not necessarily affect the structure of the protein if they happen on the intron, or if they're silent mutations like we talked about in the previous video. But sometimes you actually change the amino acid that's represented. And once the processing of messenger RNA is completed, it will exit the nucleus through the nuclear pores and go to the cytoplasm, where a ribosome will look to attach to it, initiate the process of translation.